The overall goal of this procedure is to culture mouse cardiac valves using the miniature tissue culture system, or MTCS. This is accomplished by first assembling the MTCS without the perfusion chamber and circulating the medium to precondition the system. In the second step, the mouse heart is harvested and prepared for cannulation. Next, the heart is placed in the perfusion chamber where it is cannulated for culture of the mitral or aortic valve. The perfusion chamber is then placed in the MTCS and the pump is started. Ultimately, histological analysis is used to assess the viability, morphology, and extracellular matrix distribution of the valve. Begin by using pieces of silicone tubing to make connections between the reservoir and the pump and the bubble trap, and the bubble trap and the reservoir. When all of the tubing is connected, fill the reservoir with 70% ethanol. Turn the pump on at 1 milliliter per minute flow speed and let the ethanol circulate to sterilize the tubing. After 30 minutes, empty the reservoir and pump the ethanol out of the system. Next, fill the reservoir with sterile distilled water and pump the water through the system to remove the residual alcohol. Then, pump the water out again and fill the reservoir with 45 milliliters of freshly prepared medium. Circulate the medium to